So we have a session now where we're going to be able to hear Dr. Collins presenting some of the NIH vision, followed by a couple of the IC directors, discussing through really how NIH and the various IC programs sort of fit in with the ICDA vision, or rather how ICDA fits in with the NIH vision maybe. And we are really excited to have this discussion. So the goal of the panel is to have a robust discussion around this, and I'm going to start with introducing Dr. Collins. He hardly needs any introduction. He's a physician geneticist who has a long-standing interest in human genetics. He's been instrumental in some of the early positional cloning efforts. He's been a co-lead of the Human Genome Project, and since 2009, he's been the director of NIH. So without further ado, please, Dr. Collins.
Well, thanks, everybody. It's wonderful to have a chance to come and speak to all of you. And NIH is delighted to be a host to this very significant meeting, the first gathering, the launch, as it were, of the ICDA, an organization and community about which we have great expectations and an opportunity here today and tomorrow to try to see what those might look like. So it seemed that part of my charge was to think about where this whole field of genomics is going in the future, which caused me to worry that I'd be expected to make predictions, and there is trouble afoot as soon as you try to do that. Notice this particular phrase attributed to Niels Bohr. I thought it was Yogi Berra, but apparently it really was Niels Bohr who said this. Prediction is very difficult, especially about the future. Another quote that I should have paid attention to. I always avoid prophesying beforehand because it's much better to prophesy after the event has already taken place. Winston Churchill always has the best quotes, and he really said this in 1943. So perhaps I should have paid attention. But I couldn't help as sort of planning for this, thinking back upon times where we've gathered, or some of us had conversations about where is genomics going, particularly as far as its impact on medicine. And so I pulled out this article, which probably would have been better if I'd never admitted existed, a special article in the New England Journal of Medicine 20 years ago by myself, The Medical and Societal Consequences of the Human Genome Project, and tried to make predictions about what might happen in terms of the practice of medicine as a result of what appeared to be at that point a pretty exciting set of revolutions, and indeed it was. So in there was a hypothetical case of John, a 23-year-old college graduate with a high cholesterol and who's a smoker, and some other things that are cut off at the bottom here about his history. But here was an opportunity for precision medicine, although we didn't call it that at that point, to kick in and help out John. So in my hypothetical case, what happened? Well, John, of course, had some genetic testing to see what might be useful to him in terms of practicing better health maintenance. This is a table from that particular New England Journal article. There's some made-up genes here that don't exist. There's some that do exist. But the fundamental mistake that I made, and I think everybody else probably made with me at the time, was to assume that the genetics of common disease was going to be oligogenic, not super polygenic as we now know it to be. So that in fact, we would be able to help John by simply assessing alleles at a rather short list of genes and be able to make predictions about his relative risk and his lifetime risk. And he would, of course, be immediately motivated to act upon those results, change his behavior, and live 2 decades longer than he otherwise would have. Wouldn't it be nice if it had all turned out? And by the way, this was supposed to happen by 2010. Oops. So, okay, science is a little more complicated than that, and delivering healthcare is a lot more complicated than that. So with that sort of sobering consequence, here are we now, and what should I do in terms of trying to think towards the future? I will not have any cases of anybody named John this afternoon or anything quite like that. But I do believe we are at a critical juncture, and why this meeting is so timely right now, because of the progress that has been made across a wide variety of areas of opportunity. And this is a good moment to think about what other investments should we be making. I'm going to align myself with those who think that the most useful way to have that conversation is to focus on specific projects with specific goals and specific timelines and milestones. And oh my God, that means budgets will have to be involved pretty soon too. And as the NIH director, I do have to think about the budget. Maybe you all don't, but it's rather important to keep that in track. So let me try to walk through where we've been and where we're going And I think some of what I will say and where we've been will probably echo a fair amount of what you heard from Eric Lander this morning, so perhaps I'll go through it fairly quickly. But if you don't know where you've come from, you don't know where you're going, as Maya Angelou, my favorite poet, said. So where have we come from? I guess another milestone that I was thinking about this year, in fact just this month, it was 30 years ago that the gene for cystic fibrosis was identified. This was, for those of you who were still in diapers or not born yet at that point, this was a brutal experience trying to find a 3 base pair deletion in a gene on chromosome 7 that had not previously been described before. With no human genome project, not a whole lot of internet assistance, technologies that were really quite clunky. Oh my God, all that P32 that you had to use to do almost anything. This was 5 years, maybe more than that, of hard work by a half dozen groups. Probably cost somewhere in the neighborhood of $50 to $60 million of research money, burned out a lot of graduate students and postdocs, running down blind alleys that turned out not to be it, until finally this result emerged. This was, for me, one of the big motivators to say we've got to come up with a better way to do this. So as you now encounter really critical challenges that are just too clunky and too hard, maybe that's part of what we're here to talk about. What would empower the process to make it exportable and expandable? This, of course, is now the search for a gene like this in a well-behaved autosomal recessive disease. A mediocre graduate student with access to DNA samples, a thermal cycler, and a DNA sequencer could nail this probably in the space of 2 weeks. So that's good, and that's a kind of acceleration of the research enterprise we want to empower for all the steps that are now in front of us. Of course, this did come at the time just as the Genome Project was being discussed, and I do think it was of some help to show that this was a strategy that could succeed in finding an important disease gene, but that if we really wanted to do this at scale with thousands of disease genes, it was going to take an organized effort to lay out all of the properties of the genome, to develop the technologies to sequence DNA at much faster rates, and of course that is exactly what happened, and I won't belabor it other than to say it was an amazing ride, an amazing adventure, 2,400 scientists in 6 countries, the first really big science project in biology, and one which I think will stand for the course of time as having been one of the most significant achievements of humankind about that time of our human history. By the way, at some point, people started calling everything after that the post-genome era, and I want to object against that in the strongest term. Daggone it, we were in the pre-genome era for all of human history. We finally got to the genome era. Can we just be there for a little while before we move on and say we're post all of that? So I'm going to say we're in the post-HGP era because we declared victory with a few little issues about whether we really had that complete sequence. We've continued to complete it ever since. But we are not in the post-genome era. We're in the genome era. That's what we're here to talk about. But we do want to take all of that development and figure out, for all of us who care about human health, how do we take that to the point where a scenario like what you see here is not the ultimate outcome, where somebody's really sick and the people who are trying to sort it out are throwing up their hands going, what the heck are we supposed to do with this information? NIH jumped in at that point with a lot of help from a lot of you to try to identify what were those other large-scale databases that we would really want to have for interpretation of that reference sequence. And out of that came a whole host of projects, all of which involved a lot of time and effort and organization and hard scientific work and money, only a few of which are actually listed here and many of which have been talked about at this meeting. Maybe I'll mention just a few of the significant parts of that. Obviously, the fact that we did have then this drive to do a lot more sequencing was a main driver for why the cost came down. We could argue about what it is. I think our genome centers, when they ask for money, say they do this for $850. Sometimes I wonder if they do it for a little less than that, but anyway, it's come down remarkably, and I want to see that curve drop further as soon as we can see that happen. That mythical $100 genome is out there waiting for us, and what that would do to empower research is hard to overstate. And of course, having all of that information about those rare diseases and the ability to go after them, which we couldn't do easily for CF, we've now done for 6,500 diseases, all of which we now have identified at the DNA level in a rather precise way. That's the good news. The bad news is that only for a few hundred of those do we have a therapy that we can be absolutely confident is beneficial, and many of those are far from satisfactory. So one of the things to talk about at this meeting is this huge gap between what we can diagnose and what we can treat, something that we all, as people who care about those families that are out there waiting, should pay attention to in the strongest way. So what do we need to do? Well, the themes from the post-Human Genome Project era might be broken down into these, and this is rather arbitrary. Obviously, we had to figure out not just a reference sequence, but what were common variants and the challenge of polygenes and how we were going to use that information. The HapMap project, following on the heels of the SNPs consortium, produced an initial view of that. 1,000 Genomes project added a lot to that. I don't have to tell this audience how incredibly valuable it has been to have that database, but also how limiting it is because it's mostly about Europeans. And the application of that to then do the GWAS studies and find the variants across the genome that relate to common diseases, which in 2006 was pretty much an empty diagram, has been an amazing romp forward. And thanks to the folks at EBI that just updated this very cool animation I'm about to show you. Just watch and see a little bit starting to happen 2008 and 9, and as we page forward here, several months at a time, you can see for those human chromosomes, each one of those dots being a GWAS finding. It's pretty amazing, especially in the last, year or so, and we're not done yet. And this was clearly driven by both having a very wonderful list of common variants, the ability to impute those by having done genotyping with a modest number of a few million SNPs, and the opportunity to collect lots and lots of samples so that your power was sufficient. So that has gone, we could all agree, really well. The problem, and here I'll use an example from my own research lab's efforts on type 2 diabetes, is that this turned out not to be anywhere near as straightforward. You can go back and find papers predicting that most of the variations were going to be in the coding region. We all read those and hoped they would be true. And we all know now they were not true at all, that most of these are outside the coding region in regulatory domains for diabetes. This is a summary of a group effort involving a lot of folks, including Mark McCarthy, who's right here, where almost 1 million patients with normal glucose tolerance or type 2 diabetes, SNPs across the genome, now 243 loci, and greater than 90% of those are non-coding, and of course, therefore, screaming for explanations and for ways that we could use that information to try to do a better job of prevention and treatment. So if there was ever a motivation to try to understand the functional part of the genome, this added to it, although we were interested all along. This became a really strong motivator to hurry up about it. And of course, understanding genome function is another area which has been susceptible to large-scale projects, beginning with ENCODE, to map this comprehensive catalog. The data always freely available from these enterprises. The Roadmap Epigenomics project coming along to produce additional functional data on epigenomics as well as transcriptomics, more and more human tissues and cell types. An international consortium, and it's really good this is an international meeting, and I want to keep emphasizing how critical that is for everything we talk about for the next day and a half. And GTEx, which was just presented in a very nice What You're About to See in bioRxiv view by Thule, has produced also a phenomenal amount of freely accessible information and also access to tissues for those who have additional things that you would like to do with them. All of this also making it possible for us to move from chunks of tissue to single cells, the revolution that's come along in the last few years, led again by many people in this room, so that it is possible to contemplate having a human cell atlas that tells you, in the normal state at least, what is every cell in the body actually up to as far as its gene expression and its chromatin structure. But of course, we really want to try to understand how to apply all this information in a broad way in the real world for prevention of disease and for management of chronic illness. And here the need for large cohorts, which has always been there, has emerged as an even more strongly compelling one. I know you had a whole session about that this morning, and I won't try in any way to replicate what must have been said in what I gather was a very inspiring session. I will have to give a shout out to the UK Biobank for having, yeah, Rory, that's you, for having had the vision here to figure out how to put together a cohort of this sort and to make the data accessible to all the people who had good ideas about how to use it. It is phenomenal to see the outpouring of findings that are coming forward from this. Notice, of course, the timetable, that it took a long time to get the database to that point, and some of us who are building other kinds of cohorts also recognizing that this is not something you do overnight, at least if you're not going to do it right, you need to take a little longer than that. All of this output, which are you no doubt using in your own research and seeing lots and lots of publications. That leads me to say a word about all of us, and I think we may hear more about it in the panel that follows, because Kelly Gebo, who is the Chief Medical Scientific Officer, will be on the panel. This was the U.S. effort to try to see what we could do to develop a longitudinal cohort of 1 million or more people living in the U.S., recognizing our considerable diversity. All countries are different from all countries. Ours is particularly diverse. And so the goal of this was to enroll 1 million people, with at least half of them being of racial or ethnic diversity groups, and also having diversity in socioeconomic status and geography and rural versus urban home, which is not often something that's been captured by a lot of preceding cohorts. And again, the mission here to create a database consisting of electronic health records, lots of questionnaires that are answered, lots of laboratory data, ultimately whole genome sequences, but beginning with SNP genotypes, and also having people walk around with wearable sensors and have periodic re-examinations to see what happens to them. With n equals 1 million, there's a lot of really exciting things one can learn in that way. We do treat our participants as partners. And so something that's a little different about this is we are giving back to them all of the data that we know about them to the extent that they want it. And that will include for people who want a lot of genomic information. This will, by the end of this year, result in a researcher workbench, which will be available for everybody to start looking at. Genetic information will start appearing next year, and away we go. And in terms of the enrollment so far, as of last Friday, 315,000 have started enrollment, which means you go through a variety of steps. 200,000 of those have completed all the steps, including physical measurements and biospecimens. And as you can see, the curve is going up pretty sharply. We do expect we will have full enrollment of 1 million by 2022 or 2023, but obviously you can already start doing a lot of work with a few 100,000. And if you want to join, and you happen to be somebody who lives in this country, joinallofus.org is waiting for you to come and sign up. Now, in terms of cohorts, I do think that there is an opportunity to bring together maybe an even more international way than has previously happened, all of these cohort consortiums. And so over the course of the last couple of years, this idea is being put forward, led by Terry Manolio, who's here, and Jeff Ginsburg at Duke, to try to figure out how we could, for all of these cohorts that have at least 100,000 participants, arrive at some kind of shared idea about what kind of standards to use for the data, and particularly how we could arrange for meta-analyses to be done across cohorts, because imagine the power that begins to appear here. You can see the numbers there of the number of individuals that are already enrolled or expected to be soon across the world here, and it quickly adds up to tens of millions of individuals. And you can imagine how much that will help you when you're trying to discern things that you'd like to know about in terms of populations, both in terms of how they're similar and how they're different. All of this is a vision then to create this globally connected network. And again, there's a website there. If you are not familiar with this, we'd certainly like to see this effort, which I think has now organized itself pretty effectively, to go forward and move the opportunity of sharing information. All of this, of course, requires standards that we can agree to about the genomic part of this. And I have to mention, therefore, GA4GH as a critical component of that discussion. And I'm glad that's represented here at this meeting as well. But of course, the title of this particular white paper, and maybe where we're really trying to get to, is maps, yes, but to mechanisms, yes, and to medicine, M2, M2M. So what do we need to do to get there? Maybe that's sort of what I will try to suggest to you might be a starting point of an action item list, although it's not particularly a private, personal one. It's one that I think is probably reflective of the thoughts of many of you. What's needed now? We need a complete catalog of all common variants in all populations, and not just Europeans. I gather Charles Rodemy spoke a little bit about that this morning. H3 Africa, which Charles had a big role in founding and which is supported by NIH and the Wellcome Trust, is in a good position, particularly if supported by something called the Coalition for African Research and Innovation, which is beginning to make some real steps forward to bring together African governments in support of this, not just in a colonial model, but when ownership also starts to happen. This would be a wonderful flagship project for such an effort in Africa. So strongly supportive of that, and to make sure that it's defined with specific goals. Similarly, you want to accelerate the process of whole genome sequencing. We have a lot of that data, but we need more. To identify rare variants of large effect in all populations, using the more sophisticated information we now have about which genes are tolerant of variants and which are not, in order to get a sense of what you need as far as power to look for this. And of course, if we have all of those genome sequences on very large numbers of individuals, we're going to find some really interesting genotypes, some that we didn't expect, where people seem to be homozygous loss of function for a gene we thought was critical, and somehow they seem to have reached adulthood, or at least have reached some kind of way to give a blood sample. And we will really want to understand what's the phenotype there. And this ability to do callbacks, reverse phenotyping, whatever you want to call it, is going to be critically important. And we could call that the Human Knockout Project that nature has already done, but we have to do our part of it to understand it. Just want to say, this will be critical, of course, for all these cohorts to involve permission for recontact, so that it is likely you can re-ascertain such individuals and ask them, not at their expense, but at yours, to come in for a really detailed analysis. Just putting a little thought bubble in your heads about how that might happen. In the US, we have the NIH Clinical Center. There's a picture of it there, on the NIH campus, only about 7 miles from here. And that is a research hospital that is specifically designed solely for research and all paid for because of the way in which it is set up. This would be a great place to begin this process of reverse phenotyping, as interesting genotypes appear that really need a very sophisticated analysis with lots of experts, lots of scans, lots of other kinds of phenotyping tools. Ultimately, we're going to need a much bigger network than one center, but this might be a good place to sort of pilot this as interesting genotypes first emerge and need this kind of exploration. We will need real-world testing of the value of polygenic risk scores. This is something the IHCC has been talking about. And one could imagine that these cohorts are going to be a good place to test that out as well, but it's going to have to be carefully designed so that we really have a sense of what happened. What are the metrics for success if you're going to apply this? Which diseases would you apply it for? I'd be very interested to see what people's thoughts might be about that. And of course, pharmacogenomics, which for me seems like it ought to be one of the early home runs of the genomic revolution, for the most part has not happened, for the most part because people don't have their DNA genotypes at the time the doc is writing the prescription. But if you have cohorts like all of us, for instance, we ought to be able to test that out, where the genotype's already in hand, and we can see what the results are as far as improving outcomes. According to the FDA, there's more than 100 drugs that have on the prescribing label that you really ought to know the genotype before you decide on the dose or whether this is even the right drug. But that's never really been put into practice outside of very small controlled studies. And then we got to take genome function to the next level. We need a complete catalog of functional elements at every cell type, and some of the discussions in the session just before this are clearly leading in that direction. We will talk, I guess, more tomorrow about the opportunities here for using machine learning and deep neural networks to begin to put together what is a raft of interesting data about genomics, epigenomics, transcriptomics, proteomics, metabolomics. I'm sure you can come up with some other omics. And this is a place where clearly the opportunities are legion for machine learning to step into this space. Because ultimately, maybe this is a little too bold, we'd like to know the functional consequences of a mutation in every single one of the nucleotides in the human genome. And there are some machine learning efforts that are beginning to predict that, in not maybe the whole genome, but in some pretty interesting functional subsets. And then because we are interested not just in basic science and not just in diagnostics, we also want to see what we can do about therapeutics. We need to figure out how to accelerate genomically informed drug development. Again, some nice examples in the panel just before this. For each disease, that kind of means trying to figure out how to identify and prioritize the next generation of targets that are worth investing in. And I will say it's been a very interesting experience for me working with colleagues in industry on the Accelerating Medicines Partnership, which is now 5 years along and has had exactly this goal, trying to take the deluge of GWAS data and understand how that could be applied for type 2 diabetes, for rheumatoid arthritis and lupus, for Alzheimer's disease, for Parkinson's disease, about to start one on schizophrenia, which is great to see that industry is actually willing to kick in some of the funding for that, because schizophrenia has been so long without that kind of a major push. But there are many other ways that one could contemplate going down this road. Certainly tissue chips and iPS cell systems to test effects on human cells without putting humans at risk, starting to look like a very promising area, but still not fully invested in. And let's be clear, though, I mentioned cystic fibrosis at the beginning. That was 30 years ago. It is now this year, and probably fairly soon in the near future, where we will see the small molecule approach for cystic fibrosis emerging as a really exciting success for most patients with the disease. It's taken that long. If we have 7,000 diseases to deal with that are single gene disorders, and then virtually every other condition that has polygenes that might be targetable, this is going to take a long time to scale to the point where you could say we are doing everything possible to find answers with small molecules and biologics. So just as a thought, in vivo somatic cell gene editing, which we are all, I think, exploring now, at least for thought experiments for single gene disorders, things like muscular dystrophy, for instance, maybe in the long term, this has a lot going for it. This could, after all, be something that could be scalable if you had an appropriate repertoire of vectors that could deliver your gene editor and your guide RNA, or whatever we develop later that gets better, to the right tissue safely and effectively, and you knew what your target was. Okay, that sounds like a scalable approach to going after that target, something that small molecules are a bit more challenging to do. In that regard, NIH has invested now $180 million in this somatic cell gene editing program, which aims to try to bring together all of the expertise in both designing gene editors, but also in delivery systems, to see how we could accelerate that process so that you would have a pretty decent menu of opportunities. Maybe those are viral vectors like AAV, maybe they're nanoparticles, maybe they're liposomes, but imagine if that was already in place and we were encountering a disease that needed a treatment. Maybe it's monogenic, maybe it's polygenic. Would that start to be an attractive alternative as a direction to go to test out a new kind of therapy? I think that's an interesting thing to contemplate. So just as a silly little finish here, because we're trying to be grandiose, right? This is the ICDA meeting. We're all together. We're going to make a big vision about where we want to go. Is this the fourth genomic revolution? I'm picking up on Klaus Schwab, who's the guy who organizes every year the World Economic Forum, which I have been going to for the last 30 years. And it's always interesting, although it's always a little grandiose and overblown. Klaus has this particular idea now that we have moved into the fourth industrial revolution. The first one being mechanization, steam and water power. The second one, mass production, electricity. The third one, electronic and IT systems, not that long ago, and automation. And now, cyber physical systems. Well, okay, can we copy that in some way to sort of say, why is this a moment? Why have we arrived at a point that we could say has the significance that it deserves a new thinking process about where to go? Okay, what was our first genomic revolution? I guess I'd call it the reference sequence. The second, having human variation. Okay, we're not quite done with that, but we have a pretty decent start. The third, functional genomics, also not done with that. The fourth really is M2M2M, the medical benefits. That's where I think our energies now particularly ought to be beginning to turn, recognizing that unless we build on the previous revolutions that aren't over yet, this fourth one is not going to move as quickly as it might. So with that, coming back to the problem of predictions, I'm comforted by the guy who wrote The Little Prince, who actually had a better idea about how to handle this. As for the future, your task is not to foresee it, but to enable it. That sounds like a genomics kind of motto that we could all throw our arms around, because that's really what's been happening for the last 30 years since the Genome Project started down this path of large-scale projects, carefully designed, open access to the data, just trying to enable all the bright brains on the planet to be able to go faster in the direction of making discoveries about how life works and how disease happens. And to the extent we can stick to that motto, I think we will serve the scientific community and the general population of this planet as best as we possibly can. So thank you. That's all I wanted to say.
So you take a seat, and I have the great pleasure of inviting up Dr. Bianchi, who's an MD PhD, a medical geneticist and neonatologist. She's the director of the Eunice Kennedy Shriver National Institute of Child Health and Human Development. And next up we have Dr. Kelly Gebo. She is a professor at Johns Hopkins in the Division of Infectious Diseases with a particular interest in HIV. And as Dr. Collins mentioned, she's the Chief Medical and Scientific Officer of all of us. And Dr. Gibbons, please, who is an MD PhD as well and a cardiologist. And he is the current director of the National Heart, Lung, and Blood Institute, which is also a center of excellence. And then we have Dr. Green, who I think is probably really well known in this room as the director of the National Human Genome Research Institute, NHGRI. And he has done a long-standing career within NHS. And last but not least, we have Dr. Woychik, who is the deputy director of the National, this is a mouthful for a Swinglish-speaking person, the National Institute of Environmental Health Sciences and the PI for the Mammalian Genome Research Group in the Division for Intramural Research as NIEHS. So without further ado, so I'm going to try to moderate this session, and we're like a bunch of racehorses ready to go. And I want to kick off with giving each of the IC directors an opportunity of discussing what they see as the greatest opportunities for genetics and genomics to impact disease prevention, diagnosis, and treatment in the few coming years. And I would want to start with Dr. Woychik, please, because Dr. Collins has just given a vision.
Great. So is the microphone on? Great. Okay. So NIEHS, it's in the Environmental Health Sciences Institute. So we're interested in disease mechanisms, but we're very specifically interested in understanding how do chemicals, air pollution, and things in the environment, how do they perturb the normal networks of genes and functional processes in human biology and ultimately lead to disease or deleterious health effects. So NIEHS has a strong history of doing genetics and using genetics approaches. It's, genetics is just such a powerful way of identifying disease mechanisms, in a hypothesis-free way. So we've used this in the past. And the approach that we're taking is not just about disease mechanisms, but we do gene by environment experiments, where we're interested in identifying sequence variants in individuals that would predispose to disease as a result of the exposure to pesticides or to flame retardants or air pollution or things that are likely to trigger deleterious health outcomes. And part of our reason for doing this is that ultimately we want to get to the underlying molecular mechanisms of toxicology. So when you're thinking about where are we going in the future, It's about molecular mechanisms of toxicology. We want to get away from this approach where we're actually continuing to use kind of these black box approaches where you take a bunch of animals of a given genetic background, you expose them to a chemical of different doses or cell phone radiation, and over the course of 2 years you count the lumps and the bumps and you have some pathology, and you try to correlate that. So we're not using any of our fundamental understanding of how does human biology work. So recently, in fact, Dr. Collins was a co-author on an article a few years ago to define toxicology for the 21st century. So that's where we're going to take our knowledge of how do things work in human biology and actually develop not just one or two, but potentially hundreds of different cell-based and molecular assays, and couple this with high-throughput robotics technology, similar to what pharmaceutical companies use for developing drugs. But our approach is to actually develop these same tools to look at potential toxicities. So that's kind of where we're going. So understanding molecular mechanisms in toxicology, and the only thing I would say is that if we want to do that, as I mentioned this morning, we have to continue to think about factoring in environmental exposures into the phenotyping of our cohorts. And so I'd suggest that let's start with suggesting that pediatricians start asking the parents of kids where they work and what are some of the industrial exposures that they're likely to be exposed to. So there are a number of simple things that we can do. In fact, we have, it's called the sister study, where you have sisters with, it's a breast cancer study, and there are 50,000 different individuals in that cohort. So we have a lot of experience on collecting environmental information, and we would love to have a chance to better interface with this group.
Great.
Is that what you're looking for?
That was perfect, I thought. Dr. Green, maybe you want to?
Sure, thank you. Obviously, my circumstance is a little bit different than some of my colleagues. I have the luxury of having Eric Lander start earlier in the day, and then Francis Collins just recently highlight a number of projects that NHGRI has been supporting and been intimately involved in or helping to lead, going back to the Human Genome Project. But the key message, I think, what I find very exciting about this alliance and the potential that it serves, really speaks to the general theme of not being able to do it alone. I mean, if there's not going to be one institute at NIH, it's not going to be one funding agency. The vision that's being laid out is not going to be just one country. This is going to have to be a tremendous number of different oars in the water rowing in a very coordinated fashion. This very much aligns with what NHGRI specifically is trying to do now. We're in the midst of a strategic planning process that'll yield a new strategic plan that we will publish really 13 months from now. And many of you have been involved in many events we've had to try to develop the vision for that strategic plan. And the reason it aligns so nicely is the recognition that where once upon a time NHGRI might have been responsible for many of these core beginning activities, and indeed we were, and you only have to go back 16 years ago to the year the Human Genome Project ended, and NHGRI was basically funding 90% of all of human genomics research that was being conducted by NIH. If you fast forward to today, the good news is that's no longer the case. And that's actually good. It means my colleagues at this table and other colleagues around the Institute and Center Directors table have embraced genomics. That number last fiscal year was down to 15%. We believe by next year it'll be down to about 10%. So whereas once upon a time we could speak for genomics largely at NIH because we were funding 90% of it, we're only going to be funding 10% of it or so as we enter the new decade. And what that means is that if we can develop a very effective blueprint like this alliance has the potential to do, and my colleagues up here can see where their institutes can put their oar in the water most effectively, this will be a much more synchronous and a more productive way for at least our funding agency, NIH, to be contributing to this international effort. But similarly, this will go on with other funders as we coordinate around the world. So that would be extremely helpful to us. There's almost nothing being discussed here today so far that hasn't been discussed as part of our strategic planning process. We want to define what we're going to do, but we also want to define how we can choreograph these activities with our colleagues at NIH and at other funders around the world.
So while I have you on the microphone, and
Sure.
since we're on the topic, and given your unique expertise in this, maybe you and also Dr. Collins could comment, because we have the intention, like you say, of being really an international effort. We've already talked about, potentially genotyping or sequencing 2 million people from Africa. would you mind just outlining for us and discussing your thoughts about how do you typically work with funders from across the world, and how would such collaborations happen traditionally, and what do you see?
Probably best for Francis to answer at the highest levels. I could give specific examples, but I think it's better from him.
There is a little-known organization called the Heads of International Research Organizations, HIROs, which has been meeting for the last 23 years, originally founded by Harold Varmus and George Rada, and which now involves membership from 20 funding agencies across the world, including the major supports for biomedical research, governments, as well as philanthropies. Around that table, when we meet, is about 95% of the public funding of biomedical research. That group, which is currently chaired by Jeremy Farrar of the Wellcome Trust, is intensely interested in coordinating genomics efforts. In the last meeting had presentations, one of them by Eric, and also focused on what we can do about GA4GH. They're intensely interested in this international 1,000-person cohort consortium that I mentioned, and trying to be sure that that gets catalyzed. So I think there is a pretty good framework of that sort. In addition, there's lots of bilateral collaborative efforts that NIH has benefited greatly from. I mentioned the H3 Africa effort with the Wellcome Trust. Certainly things we're doing with Canada, things we're doing with Asian countries. There's the Global Alliance for Chronic Disease, which is multiple different agencies focusing on hypertension, diabetes, on mental illness, on environmental lung disease, where we try to do joint issuances of grant applications, although each funding agency figures out how to do the reviews, but you end up with a global consortium that works together. So there's pretty good networking there. Again, want to be sure that the genomics part of this finds its way into the agendas and the brains of all those leaders. I think we're reasonably in good shape, but it could always be better. Eric, you may want to add to that.
No, I think the prototypic example, H3 Africa, keeps getting named. It is not an NIH-only project. It's done with the Wellcome Trust. It's done coordinated with the African Society of Human Genetics and other African organizations getting increasingly involved. And we would love to build on those partnerships in thinking forward of what we might do next.
Yeah. That's really useful. Thank you. So Dr. Gibbons, would you mind giving us the view from the Heart, Lung, and Blood Institute, please?
Sure. So our institute, as part of its portfolio and charge and mission, turned discovery science into the health of the nation, related to the #1 and #3 killers and promoters of death and disability. And that challenge is to really start to leapfrog from treating things on the back end after someone has a heart attack or has heart failure, earlier and earlier in that process. In the end, obviously, we would hope not to just treat diseases and sort of reduce suffering at the end stage, but actually be able to prevent or preempt and even move toward a notion where we can start to be perhaps a little bit more like our oncology colleagues, where we find a window where we can help an end organ repair and regenerate itself, such that you're not taking pills for the next 20 years, but we are actually able to stop a disease process in its tracks. In order to do that, we'd have to really have a fundamental understanding of the perturbations in human systems biology that predispose to our diseases and have that molecular understanding. So we're very excited about this alliance and its potential. I guess the other couple points I would make is that as we look at where the greatest burden of diseases that are in our portfolio, although clearly genetics are critically important, you can't help but see that the prevalence is in Kentucky and Western Virginia and in Arkansas and in Mississippi and Louisiana. And I don't know if that can be entirely explained by genetic determinants. And so as we do these complex polygenic traits, I hope this group is embracing the notion that what we've known for the last 60 years from Framingham, and that behaviors like smoking, physical activity, diet, are also important. And I always remember, can't see Sake in the audience, but I always love his diagram that shows the various determinants of high cardiovascular risk and how they're modulated by healthy lifestyle behaviors. And so I hope that we're thinking about a synthesis of multidimensional data sets. That's really the intention of what we're doing in this space with TopMed, leveraging the deep phenotyping, longitudinal follow-up of our classic cohort studies and superimposing layers of omic technologies on that, being up to about 150,000 whole genomes at this point. And then putting that in a frame where, again, we're charged with advancing the health of the entire nation and ensuring that we have an inclusive and diverse participant population, and one in which I think we may be atypical in that European ancestry is actually the minority of our data set. And I hope that that's not because of political correctness, but an intentionality to understand the molecular basis by embracing its variation throughout the human family. And then finally, I would say that we hope to take that resource and be part of this international community by creating that sandbox, if you will, that is findable, accessible, interoperable, reusable, and AI ready. And so we want to also put that data in cloud-based resources, which we're doing through Data Stage, similar to Anvil, and to create that as a resource that we hope can connect with other international data sets.
Dr. Gebo, please.
Well, one of the benefits of going second to last is that many of your ideas have also been reiterated. I think some of the things that we bring from the All of Us study is the diversity, and as Dr. Collins was mentioning, we strive to have a very high rate of diversity. We're aiming for over 75% of our participants being what we consider to be UBR, underrepresented biomedical research. And this includes racial and ethnic minorities, but also a lot of other social determinants of health, such as rural and urban status, educational level, literacy, and age. I think these are very important as we think through what are the impacts in terms of health. One of the other things I think is very important is thinking not only about genetics, but what are the social determinants of health and other types of things in the environment and other behaviors that we can modify that also impact our health outcomes along with our genetics. And I think the third thing that's very important, and in our scientific framework for the All of Us study, is thinking about wellness and resilience. So it's not only looking at an adverse outcome or a disease type, but thinking about what are the factors associated with wellness and how can we promote both physical and mental wellness as well as resilience. And I think those are things that we're hoping to look into, combining the data that we're able to get through electronic health records as well as the survey data to look with our genetics.
Thank you. And Dr. Bianchi?
Thank you. So we just released our first strategic plan in 20 years last week, so it's publicly available, and I'm here to remind you that NICHD is not just children, it's reproductive health, it's rehabilitative health, and when you talk about environment, the ultimate environment is what happened to you when you were in the womb. Everybody was in the womb at one time, everybody was connected to a placenta, and the placenta needs to be critically examined as part of GTEx. And with regard to that, we've been in discussions with NHGRI about creating a developmental GTEx that includes a little bit more depth in terms of reproduction, in terms of the placenta, and also in terms of infants, children, and fetuses that spontaneously miscarried. We really need to take a lifespan approach. I would like to challenge Eric to maybe include in the white paper another chronic condition, which is the predisposition to preterm birth. There's actually been quite a lot of studies now using 23andMe as a database. There have been genes that have been identified, and in the United States alone, 10% of the population is born prematurely. That is associated with incredible morbidity and mortality. And I would challenge you, I don't know, can anybody raise their hand if their internist ever asked them how much they weighed when they were born? I would suspect none of you. But your birth weight is incredibly important in terms of your predisposition for a number of chronic conditions. So I think it would be nice if you could include that in the white paper. We have invested as an institute $80 million into the Human Placenta Project. All of the data being acquired are publicly accessible, and there are quite a number of genomics studies there. The placenta, so many fascinating genes are expressed in the placenta, including, for example, APP. Whoever thought that APP would be expressed in the placenta? And I also want to mention that at the very same time that this workshop is going on, there's another workshop going on for INCLUDE, investigating co-occurring conditions across the lifespan to understand Down syndrome. And so you may run into some of these people tonight, but there's some at the, in the restaurant, but there are many, many thousands of people with Down syndrome who are now in research registries, and the purpose of that study, which we are one of 18 institutes involved in, is to understand both risk and resilience for common conditions like Alzheimer's disease, for example. The only other thing I wanted to mention briefly is that NICHD has had a long-standing commitment to funding clinical research networks. They've essentially been in place more or less with the same format for 30 years, and I think now is a time to rethink how those are done. There's a tremendous opportunity to link sites, to share data, and really get a sense of what's going on, for example, in obstetrical care as well as neonatal care across the United States. That doesn't mean that we're not including international. We also have a global research network, but it's a very exciting time for our institute.
Thank you so much. So one of the key things that I think that we have tried to ask all the speakers and panels today is sort of what kind of obstacles and barriers you see as the major sort of holdups at this time. And I wonder again if, maybe I can ask a few of you what you think, based on your program, what would be the biggest obstacle, and maybe if you, channeling Eric here, can sort of provide a practical solution for overcoming that. Maybe starting with Dr. Green.
Data, and data. Yeah. I mean, Gary and I were just on a phone call. We were just on a phone call, a conference call of basically an NIH council that's trying to deal strategically with data science. Yeah. And I don't even know how many different angles to make that measurement, but there is just, and this is even before we go international, even harmonizing data across institutes turns out not to be simple. The whole world of cloud computing, the whole world of consent, the whole world of international harmonization. Then we actually, even if we have all the data in front of integrating it, workforce, I don't know, what am I missing? I mean, it's like, I will tell you, and I don't know if Francis wants to add to it, there is a significant amount of energies being committed to data science at NIH. At every institute level, but also at a trans-NIH level. And I'm sure we don't have it right. We are trying very hard, but I certainly would say that if there's vision that could come out of this alliance of specific to-dos, we would love to hear those. Yeah.
Just to add to that, we are recruiting a chief data strategist, somebody who would have the vision and maybe the Silicon Valley experience to try to lead this kind of effort, which is going to be critical for our future. So that posting is out there somewhere, if you happen to be interested or know somebody who would be. The other thing I would say is all of us who are interested in seeing the international effort succeed are aware of just how many barriers pop up in that space. And I have to say one of them that has really set us back quite significantly is the GDPR, the General Data Protection Regulations, which came out of the EU, which for many of us who had long-standing collaborations between the US and academic institutions in Europe has basically brought everything to a stop until a whole phalanx of lawyers figure out how to interpret what is unfortunately a bit of a vague set of regulations. So as scientists, I hope we get a chance sometimes to weigh in on these things, but often aren't asked. This is going to be a really important issue. If we're serious, this is the International Common Disease Alliance, but we can't share data with each other. It's not going to be very successful. So please find out what's going on in your country in that kind of discussion about data protection. A lot of it I don't think is particularly well founded on benefit to people. It's some other kind of motivations, and try to see if we could avoid any more of these unnecessary obstacles that have been placed all too often without much consideration of the consequence.
Actually, the other comment that I'll make around data science is being very carefully articulating how you're going to use the data in the end, because that can influence how you collect the data. And you want to collect it around common sets of vocabularies and standards. So you're not trying to integrate a bunch of very messy, heterogeneous data, but articulate your plans. It really gets to the point, Eric, that you laid out right at the beginning. I mean, one of the successes of the Genome Project, there was a very well-defined plan. I mean, the world community knew what they could do to align themselves with this plan and contribute to the plan. I mean, now we have a lot more complex issues. So we want to think about developing plans around these individual issues that all are under the umbrella of really taking it to the next level, as Francis described. So having those clear plans articulated, you know, how can I in my individual research lab, or I in my my core lab, or my institute at the NIEHS actually work together in a way to collectively achieve a big goal.
Great. And then I'm curious also to see, you've listened into some of the discussions here and you've gotten a white paper. If you wanted to feedback, sort of if you felt that there were any key areas that we had sort of missed out on, you have the opportunity to tell us. So maybe Dr. Gebo, you want to start?
I think one of them is workforce, and making sure that we have an appropriate workforce who's trained to be able to interpret these results. One of the things that we want to do in the All of Us research program is to return our results to our participants. And when they walk into their provider's office, that the provider is able to understand what they're supposed to do with those results. And we're finding that that's not actually the case. And I think this is going to be something that we're going to have to work with, not only with healthcare providers, but really training others within the field of genetics to understand what this means, so that when a patient or a participant gets results back, we have a genetic counseling center, but this is going to have ramifications for their family members and for others as well. So I think it's making sure that we have genetic counselors and others who can help provide that information, because it's going to generate lots of stress, and that we have those resources available.
Yeah, it's a good point. I was thinking about it when Kari mentioned his death prediction for the elderly women in Iceland, that communicating that kind of information is incredibly difficult, and especially when it comes to incidental findings and so forth, how you in an ethical perspective, and I guess Bartha and Mike are going to sort of talk about that tomorrow as well. So Dr. Gibbons, do you have any sort of voids in the white paper that you want to point out?
Well, I don't know if it's a void, but I guess I would hope, I guess I'll speak as a cardiologist scientist and clearly not a geneticist, that we do consider the complex trait and the phenotyping of it. Part of the challenge in our institute is that we have categories like chronic obstructive lung disease and heart failure, in which as well as my favorite, idiopathic pulmonary fibrosis, where our ignorance is implicit, explicit in that name, and so
may as well just call it. We don't know what the hell this is.
Exactly. So, but before we do a GWAS on it, maybe we ought to do some going back and forth again in terms of probing what is this phenotype or sets of phenotypes, and I think that's another opportunity where. As a community, agreeing on common data elements and other elements of phenotyping, as well as going deep in those phenotypes. And so I hope that's another area of dialogue in addition to the ATCs and Gs.
How about you, Dr. Bianchi?
Well, I would agree that the workforce is key, because in a sense the technology and also the role of industry vastly outpaces what's going on at the medical school level or even earlier, although I have to say I think high school students in many cases are better trained for genetics and genomics than most practicing physicians. So the dilemma just increases over time, and I've certainly experienced that personally with prenatal genomic testing, but there's this rapid pace that's not going to stop. So it's like a train is approaching, and how are we going to deal with this? So we really do need some sort of urgent solution to address what is already out there and what is coming very soon in terms of patients. The other thing is, it's not a top-down situation anymore. patients have access to information, patients make choices, patients vote with their feet. So we really have to think about that in the big picture. It's not what we decide, it's how do we incorporate the participants and the patients into the promise of genomic medicine.
Dr. Collins?
Another area, the workforce, which is mentioned in the white paper, but I think is going to be just absolutely critical for the future we're all hoping of, and that is to build a deeper bench for those who are bilingual in biology and in really sophisticated computer science, because so many of the opportunities that we now can see in front of us are going to depend upon that. I mean, just listening to some of the presentations today, it's clear that's where our field is going, and yet the number of individuals who are truly facile in those joint spaces is still way short of what it needs to be for the promise to be realized. So we have to figure out how to lure all of those bright brains from Silicon Valley who are designing the next app or some other way to make money for some company, that what they really want to do is to take a pay cut and come and work in biomedicine, where the problems they get to work on are incredibly interesting and significant for the future of our species, even though, yeah, some of the other aspects may be a little different. I think there's probably a significant fraction that would get that if they knew about it. But then we have to have the training programs to be sure the biology part doesn't get missed. We've all had that experience of seeing a really sophisticated artificial intelligence analysis of a biological data set that came up with a conclusion that made absolutely no biological sense at all because of sort of something being missing there. So we have to have not monolingual but bilingual. We need more of those people, and anything we can do to figure out how to bring them on board will serve us well.
Dr. Woychik?
Actually, just a quick comment here is about complex traits. Every time I listen to a Doug Wallace talk on mitochondrial genetics, I always kind of come away, why are we studying the nuclear genome? It's all in the mitochondria. So just making sure that as we put together programs going forward, that we pay attention to the mitochondria, we pay attention to the complex mitochondrial genetics, but also I think it's becoming increasingly clear that looking at mitochondrial function, and it's not just about ATP and ROS, some of those metabolites we know are cofactors or substrates for epigenetic reactions in the nucleus. So having good metabolomic tools to evaluate the level of these within cells. So my challenge for the people working on single cell analyses is look at single cell metabolomics and then those post-translational modifications of proteins, which are critically important for human biology.
That's a great comment. So I want to take the opportunity to like the last few minutes and just open up to the floor. If anybody has any questions, please attack the microphones. I have more if nobody dares. A bit.
Don't be shy.
Oh, we have one over there.
Hi, I'm Mikhail Magitslar from GSK, and my question to the panel, how do you see bringing the pharmaceutical industry to take part of the active part of that effort?
Pharmaceutical industry. Francis talked about AMP.
Well, I can talk about the Accelerating Medicines Partnership as what I think has been a pretty good experiment. This was very much based upon the sense that industry was also clearly impressed that genomics was providing new insights into possible therapeutic pathways that they might want to invest in. But 5 years or so ago, the sense was there are just too darn many of these, and how are we supposed to prioritize? GWAS was supposed to give you a short list. It gave a huge list. And other aspects of what was possible also in a pre-competitive way seemed to be calling out for a partnership. So AMP was designed to try to bring together the scientists from both sectors around the same table to figure out what we could do together in a pre-competitive, open access way. And industry asked to pay 50-50 for the cost and funneled through the Foundation for NIH. I think that's been a pretty good experiment. It has certainly brought many of us in a circumstance where we have comfortable scientific relationships with leading R&D folks in the big pharmas that have a lot of similar interests. And this has spilled over into other things like potentially now one on gene therapy. There is one on cancer immunotherapy. It is an opportunity for that kind of shared expense and shared brainstorming to happen. I'm not saying that's the only area of opportunity, but it has worked pretty well. The downside of it is it's all big pharma. The biotechs, where a lot of exciting stuff is going on, have not been engaged in AMP, and we have to figure out other ways to build those bridges. Always, of course, challenged by intellectual property and what can be open and what has to be secret.
So just adding to this, it's not just the pharmaceutical industry, it may be the chemical industry, from the standpoint of environmental health sciences. I mean, if they can be contributing in a pre-competitive space and convincing that this does exist, they could be contributing to those fundamental insights into molecular mechanisms of toxicity. So when they put a product on the market, they have a pretty good sense that it's going to be safe. Because the last thing you want to do is, talk to Monsanto right now with glyphosate, having to deal with all of the legal ramifications of putting something on the market that may have potential toxicity.
One of the things I just wanted to raise quickly is we've talked with our participants about this and thinking about our participants as partners, and we've seen a wide variety of reactions to it. Some are very in favor, particularly people who have a specific disease and are hoping for a particular treatment or diagnostic test that could be developed. And some of our participants feel very strongly that we should not. And so I think one of the other things we have to do is educate and help them to learn about why these are really beneficial relationships and making clear and transparent what the relationship is and why it's going to be helpful moving forward.
Francis, I guess another option here is the strides and the notion that it's not, the public-private not only extends to pharma, but other equities that are interested in big data and leveraging that toward better health predictions and interventions. And so I think it's also not only about the big pharma companies, but Google and Apple and others as well.
And Amazon, yeah.
So Dr. Nuppers?
I just want to reply to Francis' comments about this GDPR. It's not as depressing as it looks. There are a lot of avenues available. Don't listen to all the lawyers, Francis. I can say that because I am one. There are many avenues. There are adequacy avenues. So in addition to this explicit specific consent, which we don't have for most of the samples, for most of the participants from the past, there's adequacy, there's public interest. You can have a binding contractual clauses. You can have a code of conduct. So there are many avenues within the GDPR that will allow us to continue international collaboration. We just have to be imaginative and dig them out and develop them.
We should talk.
And then we have one last question there. We need to stick for time today. So,
David Goldman, NIAAA. So my question is for Francis. How can we stop the new therapies from being so fantastically expensive that they could never be available to the hundreds of millions of people who need them?
Well, that's obviously a critical question for the future of all of medicine. Unfortunately, NIH doesn't have many levers to pull in that space. You may remember the experiment that was done where NIH insisted upon reasonable pricing agreements for intramural collaborations back about 30 years ago. And immediately after that, there were no more intramural collaborations because industry was not willing to make that bargain. I think what we can do is to try to move the science forward as rapidly and efficiently as possible so that the actual cost of developing a new drug and manufacturing whatever it is that has to be manufactured continues to plummet. It makes it harder and harder, therefore, for the prices to be jacked up to ridiculous levels. But that will not potentially be the only solution. There's obviously a broader discussion going on in our society about how better to handle what is currently driven by what the market would bear as opposed to oftentimes what it actually costs to get there. But we can do our part. We just shouldn't be put in the position of being asked to solve the problem ourselves with no help from anybody, because we can't. And the Bayh-Dole Act and March-In rights is not the answer to this.
I agree. You know, this may be a unique moment for that transaction between research participants and government and pharma to be reconfigured on better terms.
That would be bold.
And one last question down there.
Okay, thank you very much. So it's very exciting to hear about these opportunities, but as intramural investigator from NIH working on translational genomics of cancer, I actually didn't know about these opportunities, and I would like to know about them more and be involved in that. So maybe we also need some education for people working in NIH, that we actually have a chance to participate and use these amazing opportunities.
Well, since I'm somebody with an intramural lab, as are the other people at the table, we would agree with you. Intramural is an incredibly powerful resource. It is 11% of the NIH budget. We have, I think, in some instances, done a good job of including intramural investigators as part of some of these large-scale efforts, but not always. So, Mila, your point is well taken.
Thank you.
Thank you. So on that notion, and for everyone who's live streaming, Francis is hiring, especially from Silicon Valley. So I want to take the opportunity to thank you so much again, both for your generosity in hosting and with the organization, and also for spending your time with us here. So I'm certain everybody wants to join me and thank the panel.